And so in our work, we're now taking the human version of this protein and using directed evolution to evolve it to be more potent for regeneration. And so during evolution, this ability has been lost in humans, and we're trying to reactivate that missing activity. Hi, I'm Dan Hesselson. I am the head of the Biomedical AI Center at the Centenary Institute in Sydney, Australia. I started my education in North America. I studied in Canada and then did my PhD and postdoc in the United States. I came to Australia for a really exciting opportunity to start an independent research group in diabetes at the Garvan Institute. About 5 or 6 years ago, I moved my lab to the Centenary Institute, where I started a new direction around the directed evolution of proteins. In humans, after a heart attack, there's a scar tissue forms that compromises the function of the heart. And in some simpler organisms that we've worked on, such as the zebrafish, this regeneration process recreates a fully functional heart. And we were part of a team that discovered a protein called KLF1, which is a master regulator of this regenerative process. We found that the human version of this protein doesn't have the same regenerative activity, and so we're using directed evolution, which is a process of applying selection inside the laboratory to give proteins functions that we desire, and we're applying this to human KLF1. When we use directed evolution, we generate a lot of data, and in the past we've only used the positive results or the winners to further our research. But now with machine learning and AI, we can take all of the negative results and all of the versions of the protein that did not win the competition and learn from those to speed up our research process. We have developed a new method for evolving proteins inside living mammalian or even human cells, and so this has application to a wide range of diseases. We have projects that span heart disease, diabetes, and cancer as our main focus areas. But we think that our approach has applications across all of biomedicine. This program builds on our previous work, where we were part of a team that discovered that a particular protein is very important for regeneration in simpler organisms like the zebrafish. And so in our work, we're now taking the human version of this protein and using directed evolution to evolve it to be more potent for regeneration. And so during evolution, this ability has been lost in humans, and we're trying to reactivate that missing activity. So our work on heart regeneration has been funded by New South Wales Health, the NHMRC, and a joint venture between the NHMRC and the Japanese Medical Research Agency. In this work, we collaborate with researchers at the Victor Chang, as well as at the National Cardiac Regeneration Center in Japan. When I learned that directed evolution could be used inside of a human cell, I became very excited about all the possible medical applications of this technology. And so I've devoted my lab's efforts over the past 6 years or so to developing this approach and making it really work well inside human cells. And I am passionate about training the next generation of scientists, and my team has researchers at all levels of study, from students to postdocs who are applying directed evolution to medical problems. I grew up in Canada, and I played ice hockey from a young age, and so I still play less competitively now, but a weekly ice hockey match that keeps me energized.